So genetics, what is it? Well, it's the scientific study of heredity. And what exactly is heredity, you ask? Well, basically, it's just the passing on of physical or mental characteristics from one generation, like your parents, to you. Now ask yourself how it all started. Got an answer? No? Well, we do. Gregor Mendel was a priest living in a monastery. Also in that same monastery, he was teaching high school. Boring. But wait, he was also in charge of the monastery garden. Long story short, he took some pea plants and played around with them. Turns out he made a big discovery. He made two conclusions that have formed the basis of our current understanding of inheritance. His first conclusion was that an individual's characteristics are determined by factors that are passed from one parental generation to the next. The other conclusion he made was called the principle of dominance. The principle of dominance started that some alleles are dominant and others are recessive, and that dominant alleles will be exhibited over recessive traits. Since Mendel, scientists have concluded that the factors that are passed from one parent to offspring are called genes. Scientists have also learned that there are different forms of genes. The different forms of genes have come to be known as alleles. Now, back to this Gregor guy. He kept some pretty accurate notebooks that left him tons of data to analyze. Turns out there was a pattern he saw that explained the genetic crosses he got. He later realized the principles of probability explained it all. Probability is the likelihood that a particular event will occur. Also, due to Mendel's exactness in his data, he realized that some pea plants had the same phenotype but not the same genotype. And what do these two words mean, you ask? Well, phenotype is all the physical traits you can see with your eyes, and genotype is the actual genetic makeup. Using Punnett squares, anyone can use simple mathematic probability to predict genotype and phenotype combinations. Now, although Gregor Mendel was a very smart guy, he was still a human, and like humans, his principles had flaws. Turns out there are many exceptions to Mendel's principles. Let's take two plants and show the cross between a red flower and a white flower. The result is a pink flower because some alleles are not completely dominant over another. This means the heterozygous phenotype lies somewhere in between the two different homozygous phenotypes. Another exception is codominance. In codominance, the phenotypes expressed by both homozygous phenotypes are shown. Let's look at a chicken. In certain varieties, the allele for black feathers is codominant with the allele for white feathers. Now for something a little more complex. All of these exceptions we've discussed have dealt with genes with only two alleles, like capital X and lowercase x. Many genes, however, exist in more than two forms and for that reason are said to have multiple alleles. A good example of genes that have multiple alleles would be human blood type. The last exception is polygenic traits. This instant deals with traits controlled by more than one gene. Human skin color is a good example of this. The vast variety of skin color comes about partly because of more than 4 different genes contributing to this trait.
The cell prepares for meiosis just as it does for mitosis. But as the DNA condenses, each chromosome finds its homologous counterpart. These matching pairs of chromosomes hold equivalent genetic information, one set from mother and one from father. Next comes the first shuffle of nature's genetic deck. The two homologs trade genes in a process called crossing over. This forms new hybrid chromosomes. The spindles then place maternal and paternal chromosomes randomly on each side of midline. It is another shuffle that can lead to a host of different genetic outcomes. Finally, spindle fibers pull the homologs apart. This leaves the sister chromatids intact. With twice the amount of genetic information the cells will need, nature must cut the genetic deck in two. This second division produces 4 unique sets of chromosomes. Because they carry half the genetic information, we call these haploid cells. Many of them will mature into sperm and eggs, the reproductive cells called gametes. Mitosis begins as the long threads of DNA in the nucleus start to coil. Having already replicated, these threads emerge as the double strands we know as chromosomes. Meanwhile, protein fibers grow from the migrating centrioles, forming a latticework of spindles. For reasons scientists don't fully understand, the nuclear membrane suddenly disintegrates. With amazing accuracy, a spindle from each centriole attaches itself to each of the chromosomes. Assisted by the protein fibers, the chromosomes move center stage. In a microscopic tug of war, the spindles pull each chromatid toward opposite poles. Other spindles push against each other in a ratcheting action that stretches the cell. The chromosomes then unwind and the nuclear membranes reform. With mitosis complete, the nuclei and cytoplasm separate, creating two new cells in this ongoing cycle that sustains all life.
Thanks to studies by Alfred Hershey and Chase, we know that genes are made of DNA. DNA must be capable of storing, copying, and transmitting the genetic information in a cell. The first major role of DNA is to store information. The genes that make apples red must somehow carry the information needed to produce the red pigment. This means that these instructions must somehow be written into the apple's DNA. Secondly, DNA needs to be able to be copied. Before a cell divides, it must make a complete copy of every single one of its genes. Lastly, as Mendel's experiments have shown, genes are passed from generation to generation. This means that during cell division, DNA molecules must be carefully sorted and passed along. The loss of any DNA means the loss of valuable genetic information. Although great leaps are taken, DNA does not always replicate perfectly. When the bases aren't copied perfectly, a mutation can occur. A mutation is a heritable change in genetic information. Several types of mutations can occur and can have many different effects. Point mutations occur when one or a few nucleotides have changed. Another kind of mutation is a frameshift mutation. In a frameshift mutation, an entire strand gets messed up because everything is shifted to a spot where it's not supposed to be.
DNA contains two strands of building blocks called nucleotides, arranged like a spiral staircase. Each nucleotide includes three parts: a phosphate group, a sugar molecule, and one of four bases: adenine, guanine, cytosine, or thymine. The sugar phosphate bonds form the double backbone of the molecule, the handrails of the staircase. But we find the genetic key to DNA in the steps of this stairway, the nitrogen-containing bases. These bases link up using hydrogen bonds in a very specific way. Adenine will bond only with thymine, A to T. Cytosine only bonds with guanine, C to G. While these basic pairings never change, the order of the pairs along each strand varies greatly from one species to the next. In this elegant design, we see how nature stores the instructions to build all living things.
All of this knowledge about DNA and its structure came about due to the diligent work of devoted scientists. Some of those include Rosalind Franklin, who got the first X-ray images of DNA. There was also two guys named Watson and Crick, who built the first three-dimensional models of DNA. All in all, you can see that genetics is a very vast field of biology. Without people like Gregor Mendel, or Watson and Crick, or Rosalind Franklin, we would not be where we are today. Although we've come so far, there's still many things that we are unsure of. With time, we can only hope that there will be more advancements in the field of genetics.